Hello everyone. Today we are going to discuss about the hematopoiesis. As the word suggests, hemato, that is blood, poiesis, that is formation of different type of blood cells. Hematopoiesis is defined as the process of formation, development and differentiation of blood cells from the hematopoietic stem cell, that is HSC, which is either multipotent or pluripotent in nature. In the prenatal stage, hematopoiesis occur in the yolk sac and then transition to the spleen, liver, lymph node, and finally to the bone marrow, continue for the lifetime. Hematopoietic stem cells are a special type of cells present in the bone marrow. Their number is strictly controlled by a balanced cell division, death, and differentiation. HSCs divide and germinate daughter cells. Some daughter cells retain the stem cell characteristic of a mother, having property of self-renewing and able to give rise to all blood cells type, while other daughter cells differentiated into progenitor cells that lose their self-renewable capacity and give rise to a particular blood cell lineage. Therefore, early in the hematopoiesis, a multipotent cell differentiate along one of the two pathways, give rise to either common lymphoid progenitor cells and or common myeloid progenitor cells. Both myeloid and lymphoid lineage are engaged in dendritic cell formation. The myeloid cells consist of monocytes, eosinophils, basophils, neutrophils, macrophages, erythrocytes, platelets, megakaryocytes, etc. And lymphoid cells consist of B cells, T cells, natural killer cells, innate lymphoidal cells. The common myeloid progenitor cells can differentiate into erythrocytes, thrombocytes, granulocytes, monocytes. For the differentiation and proliferation of CFU, GEMM from HPCs, interleukins, and granulocyte macrophage colony stimulating factors are implicated. Here we can see that the pluripotent stem cell differentiated into the m- myeloid stem cell and the lymphoid stem cell. Then the progenitals occur and finally form the erythrocyte, platelets, monocyte, eosinophils, basophils, B cells, T cells, etc. in the blood and tissue. Then we will go through the process of hematopoiesis. There are different types of cell which are formed from the hematopoietic stem cell. Based on that, the cells different type of processes occur. Firstly, the erythropoiesis, which is the formation of red blood cells from the from the hematopoietic stem cell, that is called erythropoiesis. It is enhanced by decreased level of oxygen in the blood, which signals for the secretion of erythropoietin. Erythropoietin is a hormone central to the formation of the red blood cell. Erythropoiesis take 2 days on average to be completed for to form mature red blood cells from unipotential hematopoietic cells. 2 millions of erythrocytes are produced every second in our body. Hematopoietic cells determined to become red blood cells usually get smaller and more condensed as they mature until they are finally lose their nuclei. Then the process of formation of granulocyte, which is called granulopoiesis, occur. The unipotential hematopoietic cells which form a meioblast is large. It has a cytoplasm. It has a cytoplasm that is stained blue with a large nucleus. The entire process occur over a period of 2 weeks. Then we will go through the monopoiesis. The process by which monocytes are formed is termed as monopoiesis. The monoblast is committed progenitor cell found only in the bone marrow. Also, monoblast has a basophilic cytoplasm with granules. These monoblasts give rise to promonocytes, which are smaller in size with nuclei that become slightly indented before becoming monocyte. Monocytes have a kidney-shaped nuclei and can develop into a dendritic cells or macrophages. Then we will go through the lymphopoiesis. The formation of lymphocyte start their first committed progenitor cells, lymphoblast. This process is called lymphopoiesis. These cells, after maturation, are able to differentiate into either B cells, T cells, and natural killer cells. Thrombopoiesis. Thrombopoiesis is the process of formation of platelets, megakaryocytes, which are extremely large cells within the bone marrow, form platelets. The process is called thrombopoiesis. When the plasma membrane of megakaryocytes are fragmented, the origin of individual platelets take place. Thus, germination take platelets containing many granules. And on this, we stop. Thank you.